Good morning, everybody.
Morning.
Good to see you. Go ahead and type your first name or your nickname into the chat for me. So I'll have a record that you attended today. Thank you. All right, we're just at 11:20. So I'm going to go ahead and get started, and people can join in as they arrive. So today is our last meeting for the semester. It's hard to imagine, but we have come through. We've come through a very unusual semester during this pandemic. And I have certainly enjoyed working with all of you this semester, although remotely. What we're going to be doing today is talking about the vaccines that are available against the SARS-CoV2 virus, the virus that causes COVID-19. And I'm going to encourage you, as always, to take a set of notes today, because what we're talking about regarding the vaccines may show up on the exam on Friday, probably in the form of 1 or 2 bonus questions. So it would be in your best interest to jot down some notes today and have them with you during that final exam, that final lecture exam, which takes place on Friday, April 30th, this week. So a couple of housekeeping things before we begin today. Remember that today is the last day that you can hand in late work. If you have outstanding work that you have not submitted yet, I extended the time that you can submit late work for credit to tonight at midnight, and that is a fast and firm deadline. Okay, so if you want to get anything in that has not been submitted yet, and you would like to earn a few points on it, you have until midnight tonight. Okay, I've noticed that several of you have submitted a couple of things, which is wonderful. All of those points add up towards your course grade. So I'm very happy to see that some of you have handed in a couple of things that have been missing from you. I'm going to go ahead and pull up Blackboard, our weekly module, so we can take a look. That's what we're looking at right now. That's what you should see on your screen, the week of April 26th. You can see the last to-do list here at the top. So again, tonight is the last time that you can submit late work, up until midnight tonight. You also have a lecture topic this week, your final lecture topic, which is called Pathogens and How They Spread. So this lecture quiz is also due by tonight at midnight. That will be the end of the lecture material that will be covered on the final lecture exam on Friday. Remember, this lecture exam on Friday is not cumulative. It's just the next lecture exam. So it's going to cover material that we've been talking about in lecture since the last exam. And that includes Intro to Immunology, Introduction to Vaccination, Introduction to Epidemiology, and this last topic called Pathogens and How They Spread. So this lecture exam, this final lecture exam, is not cumulative, and it's going to have the same format as you've come to know through the semester. You're going to be free to use your lecture notes during the exam, but no other source materials. The exam's going to be available to you from 8 a.m. to 8 p.m. You need to get the exam submitted by 8 p.m. for full credit. And as always, you have 90 minutes to complete the exam. It will be approximately the same length as the other exams as well. And as always, if something comes up that prevents you from taking the exam on Friday, you need to immediately notify me. Send me an email and let me know. It's especially important for this exam because we are at the end of the course. We're at the end of the semester. I'm offering, if anybody has an illness that comes up or a family emergency, in other words, a reason that they can't take the exam that's considered an acceptable excuse, we will provide you with a makeup exam on Monday. So Monday is the day for the makeup exam, and then that's it. That's the end of the course for us. So you have your slides posted for the pathogens lecture as always. Here's the lecture quiz. And the final exam is not available to you yet. It won't be opened up until Friday morning. So hard to believe, but we have come to the end of our time together. I hope everybody is doing okay. I know this is a very, very busy time of the semester for everybody because all your courses are ending at the same time. So if you have any questions that come up about any of the material that's going to be on this final lecture exam, make sure to send me a message and let me know, and we'll talk through any of the material that you have had questions about. Okay, and that way you can be best prepared for this last lecture exam. Okay, so I'm going to go ahead and pull up our slides for today about the SARS-CoV2 vaccines. I'll stop periodically and ask questions, and feel free, if any questions come up, to type them right into chat. I really intend this to be more of a discussion than a lecture or a laboratory exercise. You are living in the midst of a historic event. You are living during a pandemic. This is actually the second pandemic that we've had in recent years, but this one is certainly a lot worse than the H1N1 pandemic that we all lived through in 2009. All right. So as of this morning, these are the numbers that describe the current state of our pandemic here and around the world. There have been 150 million confirmed cases of COVID-19 around the globe since the pandemic started. There have been 3.1 million deaths. In the United States, we are at 32 million confirmed cases as of today. And of course, that's an approximate number, which is why the plus sign is here. And the deaths in our country are right at about 575,000 since the pandemic started. Right now, one of the hottest spots around the globe is in India. India has reported over 1.5 million cases over just the last 4 days. So the pandemic continues to rage around the world, and as it's done since the beginning, it is flaring up in different regions across time. So since we're going to be talking about vaccines today, the first thing I want to do is just list the vaccines that are currently in use, both in the United States and around the world. Most people are aware now that there are 3 vaccines in use here in the United States. There's what we're calling the Moderna vaccine, because Moderna is the name of the company that makes it. All of these are named after their companies. The Moderna vaccine is an RNA vaccine, and we're going to talk about what that means. The Pfizer vaccine, which is also an RNA vaccine. And finally, the Johnson & Johnson vaccine, which has just recently been reintroduced into use after it was paused for several days so that some side effects could be investigated. The Johnson & Johnson, or the J&J vaccine, is actually a DNA vaccine that uses a vector to deliver the genetic material. And again, we'll talk about what that means. There are other vaccines in use around the world, just not here. For example, the company called AstraZeneca, in combination with Oxford University, has a vaccine, again, another DNA and vector vaccine that's in use, particularly in the European Union. There is a vaccine that was produced in Russia called the Sputnik vaccine. That vaccine is being used not only in Russia, but in other parts of the world. And finally, there's a vaccine that was produced in China that is in use both in China and in other parts of the world. So several different vaccines are being administered here and around the world. And while they all target the same virus, they do that using different technologies. So we said 32 million cases of COVID-19 in the United States over the past 13, 14 months. Large number of cases for one country, especially in relation to the whole population of the world, of the globe. The United States has been a place where this virus has been particularly problematic compared to other countries. Vaccination status right now varies a lot, depending on not only what country you live in, but where you live within a country. And that's very evident in our country. So if you're not familiar, the United States has a population of approximately 315, 320 million people. Now, of course, a lot of those people are children. So if you look at just the people who are eligible to be vaccinated, in other words, they are over the age of 16, 267 million people in this country are eligible to be vaccinated right now. And as of today, 142 million people have received at least one dose, because of course, some of these vaccines require 2 doses. The Johnson & Johnson vaccine only requires 1 dose. So 142 million, just over half of the population in our country who is eligible to be vaccinated, who can be vaccinated, have received at least 1 dose. 97 million people have been fully vaccinated. So again, that means if a person got the Johnson & Johnson vaccine, they got 1 shot, they're fully vaccinated. If a person got the Moderna or the Pfizer vaccine, that means they've gotten two doses. Now they are fully vaccinated. That's what that terminology means. And remember, it's not the day that a person gets the 2nd shot or the day that they get their Johnson & Johnson vaccine. It takes about 2 weeks after you get vaccinated for your body to have an opportunity to have made enough antibodies. So it's 2 weeks past your final dose. So it takes a while to get vaccinated, especially if you're being vaccinated with a product that requires 2 doses. 2 doses, 3 or even 4 weeks apart, depending on the brand of the vaccine. It takes a while. So there's been a lot of talk right now in our country about whether or not people should get vaccinated. Now, of course, the public health community and the medical community have been 100% clear on this, that every person who is eligible for vaccination, again, over the age of 16, and every person who is capable of being vaccinated, in other words, they're not allergic to some component of the vaccine or something, those people should be vaccinated. They should get the vaccine. They should get whatever vaccine is available to them, and they should get it as soon as possible. Unfortunately, as the entire pandemic has been politicized, so the vaccine effort has been politicized as well. And that is really clear when you start to look at things like where people are vaccinated and where people aren't vaccinated right now. I'm going to pull up a graphic for you now. That's what you should be seeing on your screen. You can see the name of the article is How is the COVID-19 Vaccination Campaign Going? This is an article that was published in on the New Hampshire Public Radio website. And what they put together was this graphic that.
The article.
Say again?
You can't see the article.
Okay, hold on. Let me show it again. Thank you for letting me know.
Okay, now
okay, great. So this is the article that NPR published. And if you scroll down, you see this graphic here. This represents a map of the United States with Hawaii down here and some other locations that are part of our country, as well as Alaska. And what this graphic shows you is the percentage of population within each state who have been either fully vaccinated or have had at least one dose. Okay, so as you scroll around here from state to state to state, you can see that the numbers really vary. They vary tremendously, don't they? For example, up here in New England, you see a lot of this dark color. Not in our state, we're a little bit below the other New England states. But basically from the mid-Atlantic region northward, a higher percentage of the population has been at least partially vaccinated. And that looks very different from regions down in the south central part of the United States, and even out into the southwestern part of the United States. So there is some issues right now with hesitancy behind getting vaccinated. And what the public health community is trying to do is figure out the best way to allay people's fears about the vaccine, if that's what's holding them back, or to try to convince them that the vaccine is important. And of course, as you and I know now, having talked about vaccination, there are several reasons why we vaccinate a population of people, right? There are several different reasons why a vaccination program is so important. So on this slide, I've just summarized what we talked about in lecture when we talked about vaccination. We vaccinate not just for the obvious reason, which is to protect the person who's getting the vaccine, right? But we also vaccinate to protect the community. So vaccination, particularly against viruses, it's going to protect the individual who's getting the vaccine from that viral disease. And it's also going to help the community. And it helps the community in two ways. Number one, it helps the community reach what we call herd immunity. And remember, what that term refers to is the proportion or the percentage of the population that is immune to a pathogen. And generally speaking, we like to see with an especially virulent pathogen, an especially dangerous pathogen, we would aim for 90 to 95% coverage. We want 90 to 95% of the community vaccinated or having gone through an infection and having natural immunity. Now, when you're in a pandemic situation, reaching that 90 to 95% goal is almost impossible, frankly. If you get at least 65 to 70% of the population immunized or otherwise immune, you're ahead of the game. You're in good shape. And the reason is because this herd immunity that you're developing is going to cause the amount, the physical quantity of the virus in the community to drop. Remember, viruses are obligate intracellular parasites. They cannot replicate themselves. They are not cells. They are not living things, and they cannot persist out in the environment. The only way that a virus can exist in the world is if it has susceptible hosts to get into. So a good example of this, a real example, is the smallpox virus. The smallpox virus used to cause epidemics all around the globe, including in our country. We initiated a worldwide vaccination campaign against smallpox, and we successfully reached herd immunity. Again, 90 to 95% vaccination coverage. And that virus went away. That virus is no longer on this planet, except locked away in a couple of places in vials, which is such a bad idea. But the United States keeps a small frozen store of smallpox virus, and so does Russia. I imagine China also has it, but they have never publicly admitted to having it. But my point is, it's no longer out in the world causing smallpox. And the reason it went away is because so many people were immunized, were vaccinated against it, that there was no place for the virus to go. There were no susceptible hosts left. And that the number of smallpox particles just went down and down and down until there just weren't any left. Now, smallpox is kind of a special case because humans are the only host for smallpox. This coronavirus is different. As you know, we suspect that the coronavirus came out of wildlife, perhaps a bat or a civet or some other kind of wild creature. Coronaviruses have a number of different hosts that they can infect. So it's unlikely that we will ever completely rid ourselves of this virus around the globe, but we can certainly aim to get the amount of virus down to a level where it doesn't cause disease anymore. And again, the best way to do that is through vaccination. So vaccination, particularly against viral pathogens, protects the individual, protects the person from getting the virus, helps the community reach herd immunity, and decreases the amount of virus that is circulating in the community. We also know that some vaccines, at least, have to be boosted. In other words, we don't give simply one dose, we give 2 or sometimes 3 doses across a short period of time. And as we talked about in the immunology and the vaccination lectures, The reason that we booster vaccines is because we will increase just the amount of antibodies that are in that person's body, circulating in their blood. We will also let their immune system produce better antibodies. And by better, I mean better able to bind to the viral particles and destroy them. So you get more antibodies, you get better antibodies, and you get a longer time of immune memory. There will be a longer period of time that your immune system remembers that virus, so that even years from now, you will be at least partially protected against viral infection. All right. So we're going to switch gears now and talk about the technology of these vaccines. And we're going to start with the RNA vaccines. Remember, the Moderna vaccine and the Pfizer vaccine are 2 RNA vaccines. What you're looking at on this slide is a graphic that was put out by a hospital system down in Florida. I like this graphic because it's very simple, and I think it helps people understand what this vaccine is and what it's doing in their body. You can see there's a drawing here of the needle that's going to be put in someone's arm, and they've drawn in this liquid that has little blue dots in it. And then they've blown up one of those little blue dots right here to show us. So this is an RNA vaccine. And like that name suggests, it is literally made from a piece of RNA, a piece of ribonucleic acid that is surrounded by a shell. Now, this ribonucleic acid, this RNA, was produced in a laboratory. What we did early on in the pandemic was isolate and grow some of this virus in cell cultures in BSL-4 laboratories. We then harvested the RNA from those viral particles, and we determined the sequence of the nucleotides, the bases in the RNA molecule. And with modern molecular technology, we can then build a copy, essentially, of that RNA in the laboratory. So this is a synthetic molecule that was produced in the laboratory that looks like the RNA that comes out of the SARS-CoV2 virus. And what it is surrounded by is perhaps even more interesting. These are little tiny nanoparticles of some kind of a lipid. Now, this is the part of this vaccine that is very guarded. Moderna and Pfizer are not telling anybody what that special lipid coating exactly is, because it's this special lipid coating that makes this vaccine work. If you remember from our discussions about DNA and RNA in lecture, while DNA is a very robust molecule, right? DNA can last and last and last and last. We can find DNA at crime scenes, and we can find DNA in little droplets of blood, and we can get DNA out of dinosaur bones, and it's a molecule that lasts a long time. RNA is very different. RNA is exactly the opposite. RNA is a very, very fragile molecule. It does not exist for very long. It breaks down very easily. And so it would not work to just inject RNA into somebody's arm. You have to keep it protected long enough so it can get into the cell. You also have to get it into the cell. And as you know, you can't just get into a cell. If you're a molecule, you have to have certain characteristics to be able to go across the cell membrane. So what Moderna and Pfizer have done is created these little nanoparticles of lipid. Again, we don't know exactly the formula, but some type of a lipid that is surrounding this RNA. It keeps the RNA around long enough to get it into our cells. And it's the lipid that can come up against a cell membrane and basically merge with that cell membrane so that the RNA can get into the cell. So it's ingenious. This type of a vaccine is truly ingenious. It's a very simple vaccine. It's a vaccine that's very quick to make. It only took, I know it sounds silly to say, but it only took about a year to make this vaccine. And in vaccine research and vaccine production, that's a very short amount of time. They didn't have to grow virus in host cell cultures. They didn't have to harvest it and purify it. They didn't have to do any of that. Once they knew the sequence of the bases in the RNA molecule, they were able to produce it in the laboratory and then coat it with this lipid material. So fascinating technology. Here's the one big drawback to that technology. Even with that lipid coating around the RNA, this is a vaccine that is still fragile. It still has a fragile molecule in it, the RNA. And that means it has to be kept at a very cold temperature until it's ready to be used. It has to be kept at minus 80 Celsius. Very, very cold. Now, if you go into a research laboratory or you go into a modern hospital or a big diagnostic laboratory like Quest or LabCorp, there's going to be a minus 80 fridge in there, or freezer, I should say. There's going to be a minus 80 freezer in there. But if you go into a doctor's office or you go into smaller hospitals, if you go into public health facilities around the world, you're not going to find a minus 80. They're not that common. So this vaccine has a problem in terms of how cold it needs to be kept. And that makes it difficult to use worldwide. Okay. So going back to this graphic, what they're showing us down here is a host cell, or at least just a little piece of a host cell. That gray line represents part of the membrane around a host cell. Now, if you remember from when we talked about this virus a few weeks ago in lab, we know now that this virus binds to a receptor on the cells that line our blood vessels. And remember, those are called endothelial cells. So this virus, when it exists as a viral particle, this virus normally binds to a very particular receptor on endothelial cells. The vaccine is different. The vaccine can gain entry into any cell, including the cells that are in your deltoid muscle, which is where you get vaccinated. So what happens is the membrane of any cell will interact with the lipid coating in the RNA vaccine. And those two lipid materials, the phospholipids of the membrane, the cell membrane, and the nanoparticle lipid in the vaccine, those lipids are going to merge. And that's going to allow the RNA to come into the cell. So again, a very ingenious design for a vaccine. Now, once RNA is in the cell, remember, the host cell cannot tell whether that RNA came from itself or whether it came from a virus. It doesn't have the ability to tell the difference because there is no difference. The viral RNA has the exact same bases in it as the RNA that you would find in any cell. So the cell is going to do with the RNA what it does, which is translate it. This RNA molecule is going to be translated. It's going to be used to produce protein. And because the RNA came from a virus, the proteins are going to be viral proteins. And specifically, the vaccine was designed to have the RNA needed to build the spike protein on this virus, this SARS-CoV2 virus. Once the viral proteins are produced, some of them are going to be secreted out into the extracellular environment. Others will be inserted into the membrane. We'll talk about that in just a minute. But the key thing to know here is that that triggers the immune response. Once viral proteins are out circulating, immune cells like this one here, with all of the membrane extensions drawn on it, immune cells will immediately recognize these pieces of protein as being foreign, and an immune response will be generated. Remember, the first immune response is the innate response. Remember from lecture, those sentinel cells are going to recognize that protein as being foreign. It doesn't belong in the human body. And so they will bind with it, and they will start an immune reaction to that viral protein. Now, this is another graphic which essentially shows the same thing, just in a little bit more detail. What they're showing us here with the blue line, again, is some human cell, some host cell inside of our body. And the little gray circles with the red zigzag line, those are the little particles of RNA that are in these vaccines. So you can see they've written lipid nanoparticle coating, and then mRNA strand. You can call it an RNA strand because that's really what it is. It's a piece of RNA, but it will be treated as messenger RNA by the host cell because it will be set loose into the cytoplasm. And what will happen once it's in the cytoplasm is it will be treated just like any other messenger RNA. Ribosomes, and that's what you're looking at here in blue, ribosomes will attach to it and will start to read it, will start to decode it. tRNA molecules nearby will carry in individual amino acids. Those are the little red balls. Those will be joined together as the ribosome travels along the length of the RNA. And by the time the whole RNA has been translated, you'll have a protein. And again, in these vaccines, these RNA vaccines, that protein is a spike protein. So the mRNA has the code in it to build the viral spike protein. Now, this is a very busy slide. We don't really even need to look at a lot of it, but this is what a graphic that Moderna put out. So Moderna put this particular diagram out to help people understand how the vaccine is working. And there were a couple parts of this that I thought were very effective. Again, they're showing a needle going into someone's arm, into someone's deltoid muscle. And they're showing, again, that inside that needle, there are pieces of RNA surrounded by this lipid coating. They're showing over here in this box, they're showing the particles in the vaccine coming into a cell. They've drawn this yellow ribosome and they're showing it translating the RNA in order to make viral protein. They're showing us the viral protein in blue here. you can see that some of these blue pieces of viral protein are going to actually leave the cell. The cell will secrete these. And just as we saw in one of the earlier images, sentinel immune cells are going to bind with these viral proteins and start an immune reaction. But the other thing that happens, which is really interesting, is part of these viral proteins, this spike protein, will be exposed on the surface of cells. If you remember in the immunology lecture, every person has what's called their MHC, their major histocompatibility complex. We didn't talk a whole lot about MHC, only that your MHC is different from everyone else's because it's your particular label that gets put on all of your cells. And what'll happen sometimes is a foreign material will be placed in the MHC molecule and exposed to the outside, because other immune cells, like this T cell right here, they're going to come in and they're going to see the foreign viral protein in this form. They don't react with the viral protein when it first gets secreted, but they will see it if it gets displayed like this on the outside of a host cell. So both things, both secreting the protein and displaying the protein on the surface of the host cell, will trigger immune responses. And of course, eventually what will happen is you'll have B cells as part of the adaptive immune response producing antibodies. And that's what they're showing us here in yellow. And the antibodies will bind to the viral protein if they ever encounter it and destroy it. Ultimately, that's what we're trying to do here. We're trying to stimulate the innate immune system with the vaccine, which will then, in turn, activate the adaptive immune system, which will then produce antibodies. And then if we get exposed to that virus in the future, those antibodies will be ready to go. And they will bind to the virus, the viral particles, and they will destroy it. Okay, so basic vaccine science. And what makes this vaccine so revolutionary is that it is this little piece of RNA, nothing else. It doesn't contain the SARS virus. It doesn't even contain pieces of the SARS virus other than a copy, a replica of the RNA molecule that is found inside that virus. So very revolutionary technology here with these RNA vaccines. Let's switch gears here and talk now about the other type of vaccine that's out there, which is the J&J or Johnson & Johnson vaccine, and the AstraZeneca or Oxford vaccine, as it's sometimes referred to. This first diagram was produced by the team at Oxford University that worked on the AstraZeneca vaccine. Now, this vaccine is very different from the Moderna and the Pfizer vaccines. This is not an RNA vaccine, but it's still a very modern vaccine in that it's using DNA instead of RNA. Now, I told you a minute ago that DNA is a very sturdy molecule, but you still can't just inject DNA into somebody's arm and expect it to get into that person's cells. It won't. It has no way of getting into the cell. You've got to wrap it in something. You've got to put it inside something that can interact with the membrane around the host cell and get in. Now, in this case, what they've done is something that we've done for years now with some vaccines. So this is not new technology. they took a virus that was originally out in the world called an adenovirus. Here's the word right here. There are lots and lots of adenoviruses in the world. Some of them cause things like the common cold and other respiratory diseases. This virus was harvested from the wild many years ago. It was taken into the laboratory and it was modified. And they're showing us that by showing first this blue version of this virus and then a purple version. So they've modified the virus in the laboratory. Essentially, what they've done to this adenovirus is they've thrown out its own genetic material. So basically, this, what's referred to as the ChAd-OX1, is no longer even a virus because it doesn't have any genetic material inside it. All it is the shell. It's the protein capsid and the spikes from that adenovirus, but it has no genetic material inside. So we refer to this as a viral vector. If you were to inject this into somebody's arm, it wouldn't do anything. It cannot cause disease because it has no genetic material in it. It's just the shell of an adenovirus. Now, we've been using this adenovirus vector for many years to deliver things into cells. So that part is not new. What is new is the DNA that we are now inserting into this viral vector. What the folks at Oxford did was they took the RNA out of this SARS-CoV2 virus, just like the Moderna and the Pfizer people did. They grew some virus in the laboratory early in the pandemic using a host cell culture. They purified out the RNA from the virus. And then they did what's called a reverse PCR, essentially, or what's technically called a reverse transcriptase PCR. They took an enzyme called reverse transcriptase and they put it in a test tube, essentially, with the viral RNA, and they made a DNA from it. Think about what transcription is. Transcription is a process where a cell takes a DNA and makes an RNA. What they did with this very special enzyme called reverse transcriptase is they took an RNA and they went backwards. They made a DNA from it. And again, the reason you would want to do that in terms of vaccine technology is because DNA is much more stable than RNA. They made a DNA in lab that they could then put inside this viral shell, this viral vector, and they could then deliver into somebody's body. And what makes the Oxford vaccine and the Johnson & Johnson vaccine, which is similar, what makes them important from a practical standpoint is that they don't have to be kept at such a low refrigeration freezer temperature. You don't have to keep these vaccines at -80 centigrade because they don't have RNA in them, they have DNA in them. And DNA is just a more hardy molecule. So these vaccines can be kept in regular refrigerators. And that means that they're going to be much more practical around the world. They're going to be much more easy to use and distribute around the world in places where people just don't have these -80 freezers. So what happens when this DNA molecule comes into the host cell is the host cell is going to treat it just like any other DNA. And what living cells do with DNA is they transcribe it, right? That's using the DNA to make an RNA. And then they translate the RNA. They use it to make a protein. And again, the protein that will be made, which is shown in yellow here, is the spike protein. So what's happening with the Oxford vaccine and with the Johnson & Johnson vaccine, which is shown on this slide, is one extra step, basically. Instead of putting an RNA into the host cell, they're putting a DNA in here. How do they get the DNA in? Well, they use a viral vector. They use this shell of an adenovirus. They make the DNA in the laboratory and they put it inside this viral vector. The reason they use the viral vector shell is because this thing can get into a host cell. It has its own spike proteins that can bind to a host cell and get inside. And once it's in, the DNA gets released. It will be transcribed to make an RNA and then translated to make the spike protein. So both the AstraZeneca or the Oxford vaccine and the Johnson & Johnson vaccine use DNA and viral vector technology. And those two vaccines don't have to be handled nearly as gently as those RNA vaccines do. The other benefit to using older technology like viral vector technology is that it's less expensive. And that's another really important practical consideration, isn't it? These RNA vaccines are cutting edge vaccines, the Moderna and the Pfizer, and they are expensive vaccines. They're expensive to make. And you better believe Moderna and Pfizer want to turn a nice profit on those vaccines. They worked hard to make them. So the AstraZeneca vaccine, the Johnson & Johnson vaccine are going to be less expensive because they're easier to make. That's also going to be important when you talk about sharing these vaccines with parts of the world that simply cannot afford to buy so many of these very expensive RNA vaccines. One thing that's really important to note here, though, just because the AstraZeneca vaccine and the Johnson & Johnson vaccine are using technology that's a little older, not quite as fancy and brand new, they're perfectly good vaccines. They work great. So there's been this idea that somehow everybody wants to get the best, the most modern vaccine, the RNA vaccine. But in reality, all of these vaccines are highly effective. And it shouldn't matter which vaccine anybody gets as long as they get vaccinated. But Americans are kind of funny. We're kind of funny, aren't we? We always want the best and the newest and the most fancy and the most expensive, even when another version might be just as good. So, who has questions about this? Any questions about how these vaccines are made, what they're made of, what's inside of them? As you know, the Johnson & Johnson vaccine was recently pulled off the market for a few days. It was put on a pause. And what that meant was that the government asked, the Centers for Disease Control asked that healthcare providers stop administering it for a bit. And the reason they did that is because there were 6 cases of people who had received the Johnson & Johnson vaccine and had developed significant blood clots in their body. Interestingly, all 6 of those were women. Some of them had blood clots in their lungs. That's called a pulmonary embolism. Some of them had blood clots in their legs. That's referred to as a deep vein thrombosis. One of those women died. The other women recovered or are recovering. So, out of an abundance of caution, the CDC decided, let's pause distribution of this vaccine. At that moment in time, which was now like 2 weeks ago, 6.5 million doses of that vaccine had been given, the Johnson & Johnson vaccine. 6.5 million doses had been given, and there were 6 cases of the blood clots. So that's less than 1 in a million. Now, if you remember from the vaccination lecture, we consider 1 in a million as an adverse reaction, 1 in a million to be what we call a rare and severe adverse reaction. If we see more than that, if more than 1 in a million doses causes a rare or severe reaction, we will pull a vaccine. We won't use it. That's the standard that we have set for ourselves. Now, this vaccine didn't even cause 1 in a million. It was less than that. It was 1 in over a million doses. But out of an abundance of caution, they stopped using it for a few days. And the reason they did that was because this is a brand new vaccine. This is a brand new virus. And no one wants to take any chances with it. So they stopped using it for a few days so that they could look very carefully at the medical records and the medical histories of these 6 women. And they did a lot of statistical analysis in order to try to determine whether or not it was likely the vaccine and the immune response to the vaccine that caused the clotting, or whether it was just a coincidence. Now, I don't know that they've made that determination fully yet. I can't tell you that because I haven't seen any of the reports out of that analysis. But I can tell you that they have, since that time, they've started using the Johnson & Johnson vaccine again. The committee from the Centers for Disease Control took a look at the medical histories and decided that the benefits of this vaccine still outweighed its risks. That it was more important to get people vaccinated right now with such a small risk associated with it. And because that's a vaccine that is less expensive, that you only need 1 dose for, it makes sense to keep using it. So it's been an experiment, and I think a lot of people are very uncomfortable with the idea that we are part of an experiment right now. This is a brand new virus. This SARS-CoV2 virus is brand new. It first appeared on the planet, 2 years ago. So that's brand new. We've got a brand new vaccine or brand new vaccines against it. And a lot of people are very uncomfortable with the idea of taking something that's so new. And I think we can all appreciate that it's scary. It's a scary thing. We have now given so many doses, though. Remember, even in our country, 160, whatever number I gave, 160, 170 million doses have been administered. And it's a very safe vaccination, regardless of which one you take, relatively speaking. So, as I said at the beginning, the big drive now is to find out, to try to educate people as much as possible about the benefits of getting vaccinated, to try to make them less fearful, so that they will not only come and get a vaccine, but they will become fully vaccinated. It's really interesting. I saw a story in the news about someone started polling people who had only had one of their Pfizer or one of their Moderna shots and had not gone back for the second one. So that person has some immunity, right? But they haven't had their booster shot, so they're not fully vaccinated yet. And this journalist went around and started asking people, well, why didn't you get your second shot? And most of the people said that they were afraid because they had read that after you get your second shot, you feel lousy for a couple of days. And the truth is, a lot of people do feel pretty lousy for a couple of days after they get their second shot. Remember why they feel bad, though. You feel bad because your immune system is working. Your immune system is responding to the vaccine. In a way, you should be glad that you feel bad. It's showing you that your immune system is working. But for some people, the thought of having a headache and muscle aches and fatigue for a day or two after that second vaccine, that's kind of kept them from getting the second vaccine. And that's really unfortunate because remember what the second vaccine does. It gives you more antibodies. It gives you better antibodies, and it makes the memory, the immune memory, last longer. If you don't get fully vaccinated, you are not going to be fully protected. You'll have some protection, but you're not going to have full protection if you don't get that booster shot. So yeah, it's really hard from a public health standpoint. It's very, very difficult to get a huge population of people vaccinated because there are so many different opinions. There are fears we have to deal with. There's misinformation about the vaccines going around. So we really have a public health challenge now, trying to get people educated so that they will take part in this program. And of course, it's going to be very, very important that we don't just stop with our country. It's in everyone's best interest that we get everybody vaccinated all around the world, everybody who is eligible for vaccination, because that will help us reach a global level of herd immunity. And that will make this virus start to go away. That will make this virus go down, 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 down in its levels around the world. And while it may come back, we may live with coronavirus from now on. We may experience little outbreaks of it, little local epidemics of it. It might end up being like influenza, where we expect it every winter. But at least we won't see the levels of it that we're seeing right now. And right now, we're nowhere near global herd immunity because a lot of countries can't get the vaccine right now. They simply cannot get their hands on it, which is most problematic. Any questions? Any questions about this technology or questions about what's inside the vaccines? Questions about why you need 2 doses? Questions about any of this? All right. All right, very good. Well, we're almost out of time. So I'm going to just remind everybody again that you have your final lecture quiz that's due by midnight tonight. That's the pathogens and how they spread. And if you have any work that you want me to grade for credit, you need to get that to me by tonight at midnight. No exceptions. All right. This is the final and last time that you can submit work that's late to me. Okay. Now, part of the reason that it's tonight is because I have to have the time to grade that work. Okay. So when you submit things to me, it takes a couple of days for me to get those things graded. All right. So make sure that you take the time today to get in whatever you have not submitted as best as you can, and I will grade it for you over the next few days. Okay. Remember that you have your last lecture exam on Friday. It covers immunology, vaccination, epidemiology, pathogens, and how they spread. Okay, and there will be a couple of bonus questions on it related to our discussion today. If for some reason you cannot take the exam on Friday, and remember, you have to let me know, you've got to notify me if you cannot take the exam on Friday. If you have an acceptable excuse for not taking it Friday, you can take it on Monday, but that's the only option because the course ends at this point, and I have to get your grades submitted to the registrar. Okay. Yeah, so if you've handed in some late work to me, if you've handed in now your final draft of your pathogen assignment, for example, or you've handed in your unknown to me, or you've handed in maybe some lab questions that you didn't get done, those will be graded over the next couple of days. You'll just want to keep watching. You'll want to keep watching Blackboard to see what grade you got on those assignments, okay? Okay. And if you have any questions that come up between now and Friday about any of the lecture topics that are going to be on the exam, just send me an email and we'll talk to each other and make sure that you feel comfortable with that information. Okay, make sure you get your lecture notes in shape. Make sure they're nice and complete so that you can have those next to you while you're taking that last lecture exam. Okay. All right, everybody. I'm going to let you go. Enjoy the rest of your day. I really enjoyed working with all of you this semester. Again, we had quite a challenging, we've had quite a challenging year, but I really enjoyed being able to work with you, even though we weren't able to be together in person. And I wish you all the best. Okay. All right, everybody.
Thank you. Have a good summer.
Thank you. You too. And good luck with all of your
Summer.
courses. Yeah.
Thank you.